Alright guys, let's go ahead and look at our cell theory. We know that all living things are composed of cells. All cells come from pre-existing cells. The cell is our basic unit of structure and function. That means that this is the smallest unit of living material that can carry out all activities that are necessary for life. Another thing that I wanted to highlight was that all of our cells are going to share certain characteristics. So all of our cells are going to be enclosed by plasma membranes, which was your protective and selective barrier. They're all going to contain cytosol, which is the semi-fluid substance that subcellular components are suspended in. They're all going to contain ribosomes, and they all contain genetic material. Okay, so let's look at a little bit of background. So we know that the theory that we're going to be working on and looking at is that all organisms on Earth come from a common ancestral cell from approximately about 3.5 billion years ago. Okay, so the official name of our theory is going to be the theory of endosymbiosis. So this theory basically states that our mitochondria and chloroplasts used to be independent prokaryotic cells. When they became parts of larger cells about 1.5 billion years ago is when we had eukaryotic cells emerge. Now, throughout this unit, when we look at our components and our structure, we are going to come back to this theory, and we need to be able to state the evidence that supports this theory. So when we go in and we talk about the mitochondria and the chloroplasts, we're going to make that connection in the structure of the mitochondria and chloroplast and how those structures support the theory of endosymbiosis, that the mitochondria and chloroplast used to be their own independent prokaryotic cells, and then they became parts of larger cells, which were our eukaryotic cells. So when we had our eukaryotic cells emerge, these were much more complex with internal organelles that were compartmentalized. All right, next thing we're looking at is why is it better to have tiny small cells instead of just one giant cell? So these tiny cells are going to be better than the one giant cell because we can have compartmentalization. That means that we have more specialization. Compartmentalization is going to be an important part of the organization for both our unicellular and our multicellular organisms. Okay, the second thing is the surface area to volume ratio. Now we're going to come back to this in 2.3 and look at this in more detail along with the math, but generally overall, having a larger surface area to volume ratio allows enough space to accommodate the continuous exchanges that we're going to be having going on both inside and outside. So if we look at our smaller surface area, we see that our surface area is 1 times 1 times 6, which is the 6 centimeters cubed. And then when we're looking at our volume, we've got 1 times 1 times 1, which is our 1 cubic centimeters. So this ratio is 6 to 1. So this is a larger surface area to volume ratio, meaning we have a larger amount of space available to that cell to accommodate anything that is happening. All of their functions have better functionality. Okay, you will later on have to come back in and be able to do these surface area to volume ratio and the calculations. You should be able to calculate your surface area. You should be able to calculate your volume in order to get the surface area to volume ratio. But we are going to come back to that in 2.3. So let's look at our prokaryotic versus eukaryotic. So the word pro comes from before. So prokaryotic are the ones that were the cells we had before we had eukaryotics, just like we talked about in the theory of endosymbiosis. Your prokaryotic cells are going to be a lot smaller, so these are between 1 to 10 micrometers. Our eukaryotic cells are a lot larger, between 10 to 100 micrometers. Prokaryotic are simple, whereas eukaryotic are more complex. Our prokaryotic are going to be mainly unicellular, whereas our eukaryotic are mainly multicellular. Another unique difference between the two, our prokaryotic cells do not have our membrane-bound organelles, whereas eukaryotic cells have distinct membrane-bound organelles. The only membrane we're going to see in prokaryotic is plasma membrane. Okay, so they don't have internal membranes, they don't have nuclear membranes, they don't have an endoplasmic reticulum, mitochondria, vacuoles, or any other organelles. Okay, so our genetic material for prokaryotic cells is going to be circular naked DNA, whereas the genetic material for eukaryotic is DNA that's wrapped with histone proteins into the chromosomes. Our ribosomes, prokaryotic are going to have very small ribosomes, whereas our eukaryotic are going to have larger ribosomes. So a major theme that we're going to see in biology is that function dictates form and vice versa, where form dictates your function. Since we have specialized cells in eukaryotic organisms, each of these cells has their own unique jobs. They need to complete different functions. Since they're trying to complete different functions, that means that different cells will not look alike, since their form is determined by the job they need to do. So for example, we have a nerve cell. Now, the nerve cells are responsible, or their function is to send electrical impulses. Therefore, their form is going to be long and spindly. The form matches the function, okay? So we would see that their function is dictating their form and vice versa. Now let's look at our plant cells versus animal cells. Now we do know that mostly we're going to have the same organelles and structures. However, there are certain components that we see only in plant cells and not in our animal cells. So for example, cell walls, which are your protective outer covering that's made out of cellulose, is in plant cells, and we don't see cell walls in animal cells. Another structure we're going to see in plant cells and not animal cells is chloroplasts. Your chloroplasts in plant cells contain chlorophyll, which is your light capturing pigment. This is what gives plants their unique green color. Okay, within our chlorophyll, we're going to have thylakoids and stroma. Now the thylakoids are organized into stacks, which allow them to produce more energy. Okay, so the thylakoids and that organization, so it brings back that surface area to volume ratio, is that it's allowing it to produce more energy than if it was not stacked. Okay, the other component we have in plant cells that is going to be different than animal cells is a central vacuole. Now, animal cells do have vacuoles, but the difference between the two is that plant cells, their vacuole is central. It takes up most of their cytoplasm, and when we have mature plants, we'll see that their central vacuole is going to contain the cell sap. If a plant has a full vacuole, that's a hydrated plant, whereas if they are dehydrated, they can't fill their vacuole and they wilt. When we look at our chloroplasts, we're going to see that chloroplasts have their own DNA, which resembles bacterial DNA and not eukaryotic DNA. This brings us to that theory of endosymbiosis, that chloroplasts, because they have DNA that resembles more like bacteria, which was prokaryotic, kind of proves the theory that chloroplasts at one point were prokaryotic, and then they became a part of a larger cell, so now they're a part of a plant cell, which is eukaryotic. So let's start looking at our organelles. First one we're looking at is the nucleus. Your nucleus is usually the largest organelle. This is what the component that's going to direct what's going on inside of our cell. The nucleus is also responsible for the cell's ability to reproduce, because this is our home of hereditary information, which is your DNA, which it packages into the chromosomes. And we'll come into that in a later unit. Now, within the nucleus, we are going to have our nucleolus. The nucleolus is the most visible structure that we see in the nucleus. Okay, the nucleolus is going to take instructions from DNA and synthesize ribosomal RNA. It also synthesizes proteins, and it has large and small subunits of ribosomes. Your nucleolus is going to combine proteins that it imports from the cytoplasm with the RNA that's being made in the nucleolus. Now, your nucleolus is not a membrane-bound structure. Now let's look at our ribosomes. Ribosomes are your protein factories. This is the site of protein synthesis. Their job is to manufacture proteins, and their structure is going to be a round structure made out of our two subunits. We have our large subunit and a small subunit. Now, ribosomes are found either free-floating or attached to the endoplasmic reticulum. Our free-floating ribosomes are the ones that are manufacturing proteins that are going to be used by that cell, whereas our ribosomes that are attached to the endoplasmic reticulum are manufacturing proteins that need to be exported out of the cell. Your endoplasmic reticulum is a system of channels and flattened sacs that extend into many regions of the cytoplasm. We have two types. We have our rough ER and our smooth ER. The rough ER is called rough because it's studded with ribosomes, which are producing your proteins. Your smooth ER lacks the ribosomes. It's in charge of making lipids, hormones, and steroids. It's also essential because it's detoxifying drugs and poisons from the body, so it's breaking down toxins, and it stores calcium ions in muscle cells in order to facilitate normal muscle contractions. Now, your Golgi bodies, also known as Golgi apparatus, these are your packaging and distribution centers for materials that need to go out of the cell. These are stacks of flattened sacs. They process and package substances that were produced by the rough ER, in the rough ER by the ribosomes, into little sacs that are called vesicles. And then these vesicles then carry that product out to the plasma membrane. Your Golgi bodies are also involved in the production of lysosomes, which are our cleanup crew. Lysosomes are sacs of hydrolytic enzymes. So hydrolytic basically means that they're digestive enzymes. They're going to break down old, worn-out organelles, debris, or any large ingested particles. This ability of them to break down the old, worn-out organelles is allowing the cell to continually renew itself by breaking down and recycling cell parts, and that's known as autophagy. Lysosomes are an essential component for apoptosis, which is our programmed cell death. Okay, so having apoptosis is a critical part of the development of multi-celled organisms. So our mitochondria, this is our site of cellular respiration, often known as powerhouses, because they are responsible for converting energy from our organic molecules into a useful form of energy. And the most common energy molecule we're going to see is ATP. Now, one of the key components we have to understand about our surface area to volume is also in this production of energy. Because of the folds we see in the mitochondria, it's able to make a lot more ATP. The mitochondria has an outer double membrane, and then there are an inner series of membranes that are your cristae. They are the folds, and those folds are giving us more ATP. Mitochondria contain their own DNA, which relates us back to our theory of endosymbiosis. Because they have their own DNA, kind of goes towards our theory that they were prokaryotic organisms before they became parts of a larger cell. Now, they are constantly dividing and fusing with each other's DNA in order to compensate for any defects by other mitochondria. Last one is our vacuoles. Now, the word vacuole normally means empty cavity, but we know that when we're looking at vacuoles in our cells, they are anything but empty. These are our membrane-bound structures that are used for storage. They can store water, food, waste, salt, or pigments. Now, these are large vesicles that are derived from the endoplasmic reticulum and the Golgi body. In mature plant cells, we're going to have one large central vacuole, and a couple of words that or vocabulary that we can look at with our vacuoles would be contractile vacuoles. These are where in freshwater products, these are used to pump out any excess water. We have food vacuoles, which are formed by phagocytosis of foreign material. So a quick summary of our chart in order to understand our function. Cell walls are present in all prokaryotic plant cells and animal cells. We see our plasma membrane, as we looked at our first beginning when we talked about the cell theory. Plasma membrane are present in all types of cells. So we'll see plasma membrane in our prokaryotes, our plant cells, and our animal cells. Membrane-bound organelles. So when we looked at our difference between prokaryotic and eukaryotic, we saw that prokaryotes do not have membrane-bound organelles, and eukaryotes do. So we'll see no membrane-bound organelles in prokaryotic, but we will see them in plant and animal cells. Nucleus comes back to our prokaryote, eukaryote. We do not see a nucleus in prokaryote. We do see it in plant and animal cells. Centrioles are not in our prokaryotes. They're not in our plant cells, but they are in our animal cells. Ribosomes are in all types of cells, so we'll see them in our prokaryotes, we'll see them in our plant cells, and our animal cells.